So carrying on talking about viruses and the immune system now, moving on to video number 2. So now talking about the immune response. So at the site of entry, infected cells send out chemical signals. And these chemical signals are in the form of cytokines, for example interferon, and this then renders the surrounding cells resistant to infection, and it alerts the immune system to the invasion. The very first cells surviving the scene are macrophages. These gobble up the viruses and they pump out more cytokines to attract the lymphocytes, the B and T lymphocytes, as I talked about last week. Each part of the body is protected by lymph glands, and these act as garrisons for the B and T lymphocytes. For example, the tonsils, which are placed around the entrances to the respiratory tracts. Virus-chomping macrophages make their way from the site of infection to these local lymph glands, where they display chopped up viral proteins to the B and T lymphocytes. The B and T lymphocytes circulate in the blood, constantly replenished from the blood cell factory in the bone marrow. The lymphocytes congregate in the lymph glands, waiting for the wake-up call in the form of macrophage bearing an antigen that exactly fits their unique receptor. They carry unique receptors, and they only recognise one small segment of a particular protein, of an antigen. To cover all the possible antigens, our bodies contain many B and T cells. There are so many, so that all antigens can be covered. The union of when there's the union of the receptor and the antigen, this then triggers the lymphocyte to divide rapidly. Once the B cells are galvanized into action by their specific antigen, they make antibodies, and that then obviously prevents reinfection by preventing the viruses from infecting cells by blocking their receptor for entry. The action of cells fighting invaders is counterbalanced by regulatory T cells, and these produce cytokines that diffuse the T cells' killing mechanism. And then once the microbe has been defeated, the fighting cells then die. Their response is brought to an end by the regulatory cells. Let's carry on here. Got some more notes. So where were we? Just make sure. Let's start. Sorry, I guess here we are. Right. So to more. Now moving on now to let's look at some particularly nasty viruses. Ebola. I'm sure you've all heard of Ebola because it's been in the news quite a lot. It's one of the most nasty viruses around. Now, Ebola, I'm just really interested in learning all these facts. Ebola was discovered in 1976 in the Congo, and it was named after the local Ebola River. A school teacher, by the way, I'm just a trigger warning here, because I am going to be listing some particularly horrible symptoms, so if you don't want to hear about Ebola, mute the video here, just before I say that. But for those less squeamish, who might be interested in these facts, I'll carry on talking. A school teacher developed a viral hemorrhagic fever, severe abdominal pain, vomiting and bleeding, and he died in a few days, infected 318 villagers and killed 280. Now, Ebola spreads via body fluids, blood, saliva, faeces, etc. In 2014, a health emergency was declared as Ebola spread to West Africa, and there's a 40% death rate from Ebola. The epidemic was over by January 2016. It's a zoonotic disease. It's likely that it spreads from bats. There is now a vaccine to immunise all contacts of index cases. It's an RNA virus, and it's from the filovirus family, which means thread-like, because it's like a thread. Thankfully, Ebola, although it's an incredibly serious disease with a very high death rate, is not particularly infectious. It's mainly spread through body fluids. You have to be in close contact with the person involved. It spreads more easily where there's poor hygiene. It's not airborne, so you need close contact with an infected case in order for it to be a danger, but it is a very dangerous disease. But it doesn't, thankfully, have pandemic potential, at least at this moment. Now, the coronaviruses obviously include SARS, which took off November 2002, China as atypical pneumonia. In February 2003, a doctor who had treated SARS travelled to Hong Kong, and the epidemic then spread from a hospital where he was treated and hotel guests, and it was carried to 5 more countries: Canada, Vietnam and Singapore, resulting in an epidemic. By July 2003, however, it was over, and thankfully it didn't become a pandemic. It could have done, but it was nipped in the bud. 29 countries were affected, but thankfully the SARS virus, 2002 SARS virus was because it was only infectious once symptoms developed, and that helped prevent further spread, and also the symptoms were very severe as well, which also helped them to eliminate it. Through isolation of patients and contacts. It was spread from live animal markets. The natural reservoir was identified as a Chinese horseshoe bat, which was then transferred to other animals in crowded cages. Why they don't ban these animal markets is anyone's guess. They should do, in my opinion. Another severe disease that has gained much publicity recently is the Middle East Respiratory Syndrome coronavirus, known as MERS-CoV. That's very, very similar to SARS-CoV. And that occurred the main outbreak there was Saudi Arabia, 2012, a small outbreak of a SARS-like illness. But since 2012, it's caused many small outbreaks in the Middle East. The overall death rate is 36%, but no sustained community transmission as yet, thankfully, so it doesn't yet have pandemic status. It does not transmit easily between humans. Camels are the major reservoir of the virus. The next virus that has pandemic potential, of course, is flu. This regularly mutates, which is known as antigenic drift, which is a slow accumulation of mutations over time, hence why we have a new flu vaccine every year. Now, there are 3 flu strains. 3 main flu strains are A, B and C, but only flu A is zoonotic. Flu A can undergo genetic recombination or antigenic shift that would then result in an entirely new strain of flu, and that can then cause a pandemic, which is a major genetic change with the help of wild birds. The natural hosts of flu A are ducks, but it can also infect poultry, pigs, horses and cats. Flu A replicates in the guts of wild birds and is excreted in their feces, causing no symptoms in the wild birds that carry it. But like I say, it can be spread to domestic poultry and is a major cause of death among domestic poultry. It's an RNA genome with flu is an RNA genome with 8 genes that are segmented. Instead of being a continuous RNA chain, each gene forms a separate strand. There are 16 different H and 9 different N genes, all of which can be found in all combinations in bird flu viruses. Because there are separate RNA strands, on occasions they become mixed up. Because it's segmented, it increases again the risk of a mutation. And this event can become recombined, which is a mixing of virus strains, and sometimes there's a new strain that can then jump to humans, resulting in a potential pandemic. If 2 flu A viruses with different H and or N genes infect a single cell, the offspring will carry varying combinations of genes from the 2 viruses. There have been 5 flu pandemics in the last 100 years. The 1918 one was the biggest one, of course, that we're most familiar with, and in the 1918 flu pandemic, all 8 genes came from birds. The Asian flu pandemic of 1957 acquired 3 new genes, H and N from birds. For Hong Kong flu 1968, 2 new genes from ducks. And then there was also the Russian flu of 1977, and of course the 2009 swine flu. Flu viruses are trying to pronounce this word correctly. Paramyxoviruses. The H hemagglutinin and N neuraminidase genes are the most important in stimulating host immunity. You know, when we have